Thank you for the introduction. I also thank Jay and James for the kind invitation to this wonderful symposium. Today, I'm going to talk about a recent allogeneic transplantation study of iPS cell-derived cardiomyocytes in non-human primate first. And in the second half of the talk, I'm going to introduce our recent approach to control post-transplant arrhythmia, which is, in my opinion, one of the major obstacles to use pluripotent stem cell-derived cardiomyocytes in clinic. Several studies have shown that human pluripotent stem cell-derived cardiomyocytes can regenerate injured heart in xenotransplantation model. This is one of the examples. How can I use this? This pointer? No. Where is the pointer? Oh, no pointer. Excuse me. And this is one of the examples when we transplanted human ESL-derived cardiomyocytes into injured guinea pig heart. The cell survived and improved, and cardiac contractile function was improved. But so far, most of the studies using pluripotent stem cell-derived cardiomyocytes were performed in xenotransplantation model, transplanting human cells into animal models. But we really don't know the immune response when the cells are transplanted into the same species. So we performed allogeneic transplantation study using monkey model. To minimize immune rejection following transplantation, we established MHC-matched transplantation system. Here, we generated iPS cells from MHC homozygous monkey and differentiated them into cardiomyocytes. And recipient animals are MHC heterozygous, but either of the haplotypes is identical to donor MHC. And this is the protocol for MHC-matched transplantation study. A myocardial infarction model was produced 2 weeks before cell transplantation, and animals were treated with methylprednisolone and tacrolimus, and either iPS cell-derived cardiomyocytes or just vehicle were transplanted into total 10 animals, and animals were monitored for 12 weeks. And cardiac contractile function was evaluated by micro-CT as well as echocardiography. And cardiac arrhythmias were monitored for with Holter ECG at every 2 weeks. And animals were sacrificed 12 weeks post-transplantation. And graft cardiomyocytes were imaged by GCAMP, which I'll show you this later, and the histological analysis was performed. I'll show you animal surgeries in this movie. This is a monkey heart. Myocardial infarction model was produced by ischemia reperfusion injury. First, left coronary artery shown here was sutured. Then polyethylene tubing, shown here, was thread through the suture and put another tubing on the first one and tied off the suture. And now you can see this area be exposed to ischemia. And after 3 hours of induction of the ischemia, the heart was reperfused by just removing the tubings. And then 2 weeks after the induction of myocardial infarction, 400 million iPS cell-derived cardiomyocytes were injected directly into the infarcted myocardium and surrounding area. So we analyzed grafted cardiomyocytes by histology 12 weeks post-transplantation. You can see a substantial amount of grafted tissue labeled with GFP survived in the anterior part of left ventricle. The graft cells were, most of the graft cells are GFP cardiac troponin T positive cardiomyocytes. And the next pair of pictures shows clear sarcomere structure identified by another cardiac marker alpha actinin in the graft area. The graft area was very vascularized, identified by CD31, but all of the CD31 positive cells are negative for GFP, suggesting these are derived from host tissue. The expression of gap junction protein connexin 43 was relatively weak in the graft area, consistent with our previous work. And to evaluate cardiac contractile function, we performed a micro-CT analysis. The first movie shows base level of contraction. There's no big difference in contraction in this level. When we see the apex level, the recipients of iPSC-derived cardiomyocyte showed better ejection fraction compared to PSC vehicle. When you see a longitudinal view, you can see the same difference. This slide shows quantitative analysis of ejection fraction and fractional shortening. Ejection fraction before cell transplantation was not different between recipients of vehicle and iPSC-derived cardiomyocytes. The ejection fraction at 4 weeks or 12 weeks post-transplantation showed recipients of iPSC-derived cardiomyocytes showed better ejection fraction. Echocardiography revealed the same trend in fractional shortening as in ejection fraction. Next, we try to confirm if transplanted cardiomyocytes electrically couple with host cardiomyocytes. To visualize contraction of cardiomyocytes, we generated iPS cells expressing fluorescent calcium indicator, GCAMP. You can see cardiomyocytes flashing in sync with their contraction. And I show you how we can see host graft electrical integration. This is a monkey heart transplanted with GCAMP expressing cardiomyocytes 12 weeks ago. And cardiopressure solution was injected through ascending aorta. Then the heart gradually stopped beating. After stop beating, the heart was transported from the vivarium to the lab. And in the lab, the heart was reperfused with tidal solution. Then the heart started beating again. The beating was visualized with Langendorf apparatus. This is our Langendorf system. You can see the beating heart as well as ECG electrodes. And graft cardiomyocytes visualized with GCAMP. And this is the GCAMP imaging. You can see multiple graft area flashing. And flashing cycle is in sync with the ECG, indicating host graft electrical integration. The graft cardiomyocytes keep coupling even after the heart rate was increased by electrical stimulation. And this slide summarizes the movie. There were 3 graft areas identified by GCAMP imaging. The 3 color traces in the lower panel represent GCAMP transients, and GCAMP transients are always in sync with the ECG. Finally, I'd like to show you the analysis of Holter ECG. there are ventricular tachycardia, VT, shown here. The right graph shows the incidence fraction of animals showing VT. As you can see, the incidence of VT was significantly increased in the recipients of iPS cell-derived cardiomyocytes after cell transplantation. The right bottom graph shows fraction of animals showing a sustained VT, which was sustained more than 30 seconds. The sustained VT was observed only in the recipients of cardiomyocytes. The left bottom graph shows duration of VT in each recipient. The duration peaked in 7 days or 14 days after transplantation and gradually decreased thereafter. Now, I'd like to summarize this first of my talk. iPS cell-derived cardiomyocytes survived in infected heart for at least 12 weeks following the MHC-matched allogeneic transplantation. And graft cardiomyocytes electrically coupled with host cardiomyocytes and improved cardiac contractile function. Also, transient and transplantation of iPS cell-derived cardiomyocytes significantly increased the incidence of non-lethal VT. An important question is the mechanism responsible for the post-transplant VT. Just last year, Chuck Marie group reported a similar VT in non-human primate. The VT originated from a graft area and has no reentrant pathway, and VT was sustained even after the overdrive pacing or electrocardioversion, suggesting VT was caused by ectopic pacing by graft cardiomyocytes. Another important question is related to post-transplant VT is why the VT was transient, peaked in 2 weeks, and decreased thereafter. We hypothesized that the characteristics of graft cardiomyocytes changed in vivo environment. So to make sure this hypothesis, we performed a following experiment. So 20 million human iPS cell-derived cardiomyocytes were injected into a thymic rat heart. And animals were sacrificed in 2 weeks, 4 weeks, or 12 weeks post-transplantation for histological analysis. First, we examined electrical characteristics of human iPS cell-derived cardiomyocytes in vitro. The cardiomyocytes showed two distinct action potential patterns, 9% of nodal-like cells and 91% of ventricular-like cells. As expected, nodal-like cells beaten faster than ventricular cardiomyocytes. When we saw nodal marker SHOX2, there were SHOX2 positive and negative cardiomyocytes. We quantified the number of SHOX2 positive graft cells in vivo. Here, SHOX2 was shown in red, and graft cells were identified by human lamin, which is in green. As you can see, the fraction of SHOX2 positive graft cells was decreased in the animals sacrificed 12 weeks post-transplantation compared to 2 weeks or 4 weeks post-transplantation. Next, we examined the expression of matured ventricular marker MLC2V in the graft. Here, MLC2A is shown in red, and MLC2V is shown in green. As expected, the expression of MLC2B was increased over time after cell transplantation. So approximately 10% of iPS cell-derived cardiomyocytes showed nodal-like phenotype in our cell preparation. Nodal cells disappeared over time in vivo, and most of the graft cells were MLC2V matured ventricular cardiomyocytes at 12 weeks post-transplantation. So collectively, these findings may become a good reason for transient post-transplant VT. So our next hypothesis is that a transplantation of matured ventricular cardiomyocytes will not induce post-transplant ventricular arrhythmia. So we could successfully generate pure ventricular cardiomyocytes in collaboration with Dr. Fukuda as a Japanese team. And these cells are express for actinin as well as ventricular marker MLC2B. When we see action potential of these cells, the cells shows a ventricular phenotype. So we studied the pilot experiment transplanting human ventricular cardiomyocytes into monkey model of myocardial infarction. This is a protocol. Myocardial infarction model was produced by ischemia reperfusion injury 2 weeks before cell transplantation. And animals were treated with immunosuppressants and monitored for 4 weeks with Holter ECG, and animals were sacrificed for histological analysis. Histological analysis at 4 weeks post-transplantation revealed successful engraftment of ventricular cardiomyocytes. As you can see, the graft cells express human specific troponin I in the sky area. Unfortunately, we did see some VT, but the incidence or duration of VT was substantially decreased in this specific animal. And after 2 weeks of transplantation, there was no VT at all. In summary, human ventricular cardiomyocytes survived and remuscularized the infarcted monkey heart. Purification of matured ventricular cardiomyocytes may give rise to control post-transplant arrhythmia. Thank you very much for your attention.
This is a beautiful study, very important. In order to have the graft override recipient heart, the EP will have an ectopic center. It must be a big graft. So what is your transplantation cell number and engraft rate?
Okay. In the first study, I transplanted 400 million iPS cell-derived cardiomyocytes. And the second study, which involves with ventricular cardiomyocytes into monkey, we transplanted, if my memory is correct, 20 million cells.
So for 20 gram left ventricle, 400 million is a lot of cells. That's why you have a big graft. So the graft rate, do you know the graft rate, 4 weeks or 12 weeks after the transplantation?
What do you mean, graft rate? Survival rate?
Survival cells, 6 weeks or 12 weeks after the transplantation. Do you know?
I don't know exactly.
Great talk, Yuji. Your rat time course experiment, you characterized the myocytes well. What about the connections between hosts, connexin 43, 45?
Oh.
Did that change over time?
We are still under investigation, so we need more histological analysis. But we are doing right now. I don't know right now. Sorry.
Okay.
Hi, great talk. So I've seen that also in Michael's talk and Chuck Murray's talk and your talk. So the arrhythmias that you're seeing and you're saying they're ectopic are running about 200, even 250 beats. So in vitro, we never see cardiomyocytes go more than even 60 beats a minute. So having these rhythms of 200, 250, it's kind of really high rhythm for the human cells, just spontaneously fire. Would you know any reasons for why would that be the case?
So at least in vitro, the human-derived cardiomyocytes are beat more than 200 million. That's possible reason.
I mean, 200 beats a minute. So, for example, you have 4 or 5 hertz spontaneous activity really coming from the ectopic side in the heart, or from human cells.
Mm-hmm.
So that's a really high rhythm, right? So it's so I'm curious, do you have any ideas why would you have such a high spontaneously beating or ectopic rate from a human graft when in vitro we cannot see, rarely see those that will reach even 2 hertz?
So we did see such high beating rate in vitro. So
you're seeing like 5 hertz in vitro rate?
You're right.
What happens to the cells with the nodal-like phenotype? Do they die or do they mature into the
That's a good question. We're doing several experiments to show. So the short answer is I don't know yet, but I don't know. So I don't know.
So for the cells that, even though they're purified ventricular ones, some of them, if they're not all fully mature, can still express a lot of the funny current channels. And so prior to implantation, even though they're not pacemaker-like, have you done patch clamp on lots of cells to see if there's any spontaneous depolarization residual activity still left in the ventricular ones?
Actually, we haven't performed a precise investigation about electrical analysis of in vitro experiments.
I'll ask you one last question. Do you give anti-arrhythmic drugs in your monkey model?
No.
No more questions. I'll close the session and we'll return at 10:35 for the next session. Thanks to all the great speakers.
Thank you.